Genetic disorders by chromosome. Certain genetic disorders are associated with mutations or deletions on specific chromosomes. Chromosome 3 is linked to von Hippel-Lindau disease and renal cell carcinoma. Chromosome 4 is associated with autosomal dominant polycystic kidney disease caused by PKD2 mutations, achondroplasia, and Huntington disease. Chromosome 5 is linked to Cri-du-Chat syndrome and familial adenomatous polyposis. Chromosome 6 is associated with hereditary hemochromatosis due to HFE gene mutations. Chromosome 7 is linked to Williams syndrome and cystic fibrosis. Chromosome 9 is associated with Friedreich ataxia and tuberous sclerosis caused by TSC1 mutations. Chromosome 11 is linked to Wilms tumor, beta-globin gene defects such as sickle cell disease and beta thalassemia, and multiple endocrine neoplasia type 1. Chromosome 13 is associated with Patau syndrome, Wilson disease, retinoblastoma caused by RB1 mutations, and BRCA2 gene mutations. Chromosome 15 is linked to Prader-Willi syndrome, Angelman syndrome, and Marfan syndrome. Chromosome 16 is associated with autosomal dominant polycystic kidney disease due to PKD1 mutations, alpha-globin gene defects such as alpha thalassemia, and tuberous sclerosis caused by TSC2 mutations. Chromosome 17 is linked to neurofibromatosis type 1, BRCA1 mutations, and TP53 mutations that cause Li-Fraumeni syndrome. Chromosome 18 is associated with Edwards syndrome. Chromosome 21 is associated with Down syndrome. Chromosome 22 is linked to neurofibromatosis type 2 and DeGeorge syndrome, also known as 22q11 deletion syndrome. The X chromosome is associated with Fragile X syndrome, Turner syndrome with a 45 XO karyotype, X-linked agammaglobulinemia, and Klinefelter syndrome with a 47 XXY karyotype. Robertsonian translocation. A Robertsonian translocation is a chromosomal translocation that commonly involves chromosome pairs 21, 22, 13, 14, and 15. It is one of the most common types of translocation. This occurs when the long arms of two acrocentric chromosomes, those with centromeres near their ends, fuse at the centromere, and the two short arms are lost. In a balanced translocation, there is no net gain or loss of significant genetic material, so the individual usually has no abnormal phenotype. In contrast, an unbalanced translocation results in missing or extra genes, which can cause miscarriage, stillbirth, or chromosomal imbalance. Clinical examples include Down syndrome and Patau syndrome. Cri-du-Chat syndrome. Cri-du-Chat syndrome, also known as cry of the cat, is caused by a congenital deletion on the short arm of chromosome 5, written as 46, XX, or XY, 5p-. Clinical findings include microcephaly, moderate to severe intellectual disability, a characteristic high-pitched crying sound, epicanthal folds near the eyes, and cardiac abnormalities such as ventricular septal defects. A mnemonic is, "I cry when I am very sad," which refers to ventricular septal defect. Williams syndrome. Williams syndrome is caused by a congenital microdeletion of the long arm of chromosome 7, which includes the elastin gene. Patients have a distinctive elfin facial appearance, intellectual disability, hypercalcemia, and well-developed verbal skills. They are often described as extremely friendly with strangers. Cardiovascular problems are common, including supravalvular aortic stenosis, pulmonary artery stenosis, and renal artery stenosis.